So welcome everybody to the afternoon breakout sessions. It's on microbiomes for the bioeconomy, hype or hope. And we had already very good discussion and talks in the morning session. Now again, we will have two impulse talks, and then we'll have a moderated discussion on gaps and needs related to microbiome-based RNA policies and regulations. But now let's move on with our talks. Our first speaker is Yolanda Sanz from the Institute of Agrochemistry and Food Technology of the National Research Council in Spain, and her talk is on opportunities and regulatory challenges of microbiome innovations. The floor is yours, Yolanda.
Thanks so much, Angela. Can you see the screen?
Yes, but not yet. Yeah, perfect.
Okay. It's up. Well, good morning, good afternoon, everybody, and thanks so much, Angela, for the invitation to participate in this session. While talking about opportunities offered by microbiomes, I'm going to focus on two major challenges where microbiomes could be of help. First of all, one of these challenges is to provide sufficient nutritious and healthy food for reducing the increased rates of malnutrition, obesity, and non-communicable diseases. And also another very important challenge is to do so in the context of a sustainable food production system to protect the health of the planet also, and counteract the adverse effects of anthropogenic actions and the climate change in a win-to-win fashion. Considering that a poor diet is a key determinant of malnutrition and non-communicable diseases, and also that the reductions in intakes of whole grain cereals, fruits and vegetables, it's obvious that practice in agriculture are going to be very, very important to address this major challenge that is directly related to human health. We also know that there are many factors that determine malnutrition in all forms, not only undernutrition, but also overweight and obesity that is gaining much more importance all over the world. This depends not only of environmental factors and the food production system and our economical structure and food distribution, but also on biological factors, of which I would like to focus on the role of our gut microbiome, that is a biological factor neglected so far and directly involved in both undernutrition and overnutrition. That in fact is affecting not only developed countries, but also low and middle income countries, since in fact currently half of the overweight children under 5 years old live in Asia and a quarter in Africa. During the last years, we have investigated thoroughly into the roles of the gut microbiome in humans mainly, and we realized that diet and gut microbes interact in many ways. Diet modulates the composition and structure of the gut microbiome, but at the same time, the microbiome of each individual modifies the processing and digestion of food components, generating different bioactives that could affect different organs and systems positively or in an adverse manner. So the quality of the food we eat and the diet we eat does not depend only on its composition, but also on the activity of our gut microbes in processing this food and can change the energy value and the dietary health effects of the food we eat, changing completely the concept of nutrition. In fact, it's considered that through the changes associated to the industrialization process and our lifestyle change, we have changed the incidence of both non-communicable diseases that has started increasing, and we have also caused an important damage in the biological diversity, affecting the human gut microbiota, but also the environmental microbes. In the context of the human gut, we have learned from the recent years that we can change the situation through dietary changes, permanent dietary changes, and also through reintroduction of a species we have lost during generations. But similar approaches can be applied to soil and plant microbiomes. If we compare the functions of the human gut microbiome with the functions of the rhizosphere microbiome, we can see many similarities. For example, in both cases, the microbiomes exchange nutrients with their host and improve the biological competency of both organisms. For example, in the case of the rhizosphere microbiome, the microbes contribute to the uptake of phosphorus, to nitrogen fixation, to the degradation of recalcitrant organic matter, and in turn, roots exudates provide carbon source. On the other hand, both microbiomes contribute to protect the host against the colonization by adverse organisms, for example, competing for nutrients, producing inhibitory substances like antibiotics and lytic enzymes. And in both cases, the microbes also contribute to the function of the immune system and protect the host through immune mechanisms against the invasion of infectious agents. In the case of soil microbes, it's known that those stimulate systemically the defense capacity of the plant for developing a fast response in face with hazards. So in principle, we can think of similar applications coming from the use of microbiomes in both ecosystems. For example, increased resilience and healthy adaptation to biotic and abiotic stress in the case of plants, increasing productivity, reducing the use of pesticides or fertilizers, and ensuring safety. To use also the plant and soil microbiomes for diagnosis and for predictions, and also for improving productivity yields and the nutritional quality of the food suite. So I think that we have a great opportunity of using this microbiome momentum for transforming the food system and also improving nutrition, exploiting the best performing soil and plant microbes to support a sustainable agriculture and increasing productivity in the way we need. Impulse agricultural practice that increase provision of essential nutrients and at the same time carbohydrate structures that are accessible to our gut microbes. In this way, we will be able to combat more effectively malnutrition in all forms, as I say, and also assist in healing microbiome deviations resulting for Westernized diet associated with increase in non-communicable diseases related to obesity. We have a great opportunity to reformulate also the definition of the nutritional value of foods, considering also the way gut microbes impact dietary health effects through the processing of these foods, and overall make a definitive change on how food systems are conceived and interconnected to both human health and the health of the planet. But well, regarding the challenge in the regulatory path for authorization of this type of products, and in this case, I'm going to focus on plant protection products where we could include strategies to modulate the microbiomes of plant and soils. So far, plant protection products have already included microorganisms, but most of them are single strains, and those involved in this field perceive that the requirements for the approval of these products is going to be more complicated due to the fact that they will require the same evaluations that they are doing for single strains for multiple strains. From my point of view, the process in this part is quite straightforward in the sense that guidance are already established. It's obvious that if we include more microbes, we will introduce complexity in the process. since for example, the characterization of every strain in terms of taxonomy and possible risks requires whole genome sequence for analyzing the presence of antimicrobial resistant genes, virulent factors, possible pathways generating metabolic products that could be adverse. But the process and the methodology are well established through the guidance I'm showing you in the slide. Also, EFSA has recently published a statement on how to perform whole genome sequence and report on the analysis done and the results. From my point of view, the complexity is more related to the fact that now we will have to prove the efficacy of complex communities, and the effects will depend not only directly of one strain, but of the interactions between the different strains and also the interactions with the ecosystem. In this context, with lack of standards for using omics data and best practice, as we discussed in previous meetings as well, and with lack of study models that can reproduce correctly the complexity you see in the fields. And there are similar limitations when we talk about similar approaches applied to other ecosystems, and referring to the human gut and the products we are thinking of developing there, the restrictions and complexities are very, very similar. I think that even though we have some challenge to address in this field, we have a great opportunity to improve both productivity in the agriculture system as well as to provide healthier foods to everybody in this context, taking this great opportunity of innovating in the field. And well, that's I wanted to tell you in this short talk. Thanks for your attention, and if you have questions, I will be happy to address them.
Thank you, Yolanda, for the great talk. Very stimulating. And I ask again everybody to think already about questions and to put them in the chat. We will pick them up later on in our discussion. So then we will move to our next speaker, Emmanuelle Maguin from INRA in France, and her talk is on towards and harmonized food systems, microbiome research and innovation strategic agenda, gaps and opportunities. Emmanuelle, the floor is yours.
Yes, let me share the screen. Do you see the presentation?
Yes, it's perfect. Yeah.
Okay, thank you. Thank you, Angela, for this introduction. I'm going to talk about this harmonized food systems, microbiome research and innovation strategic agenda that we are developing in the framework of the Microbiome Support Project. So I guess we all know that microbiomes and microbes are everywhere on Earth, and they are, of course, present in the whole food systems, living in close interaction with various environments, with complex food matrices, and living in symbiosis with plants, animals, and humans. And this leads or is explained in the holobiont concept. Microbiome science is a young science, but it's already highlighted many positive services that the microbiomes are giving to humankind and for the health of the planet. And those services are also very important for food system. And in fact, microbiome services and microbiome appears now are enablers for the transformation of food system toward more sustainability and resilience. For example, they can help to reduce antimicrobial use in the food system, but also inorganic fertilizers and pesticides. They are improving soil carbon sequestration, but also nitrogen and phosphorus cycle. They improve waste treatment and even help to develop new paths to valorize to upscale waste. And they are really changing our vision of medicine and of prevention and treatment of both infectious diseases, but also non-communicable diseases. Oh, I cannot change my slide. So those services are, of course, contributing to Sustainable Development Goal, to the European Green Deal, especially to the farm to fork strategy, and to bioeconomy. Those microbiome services are also stimulating and shaping new markets. And also my numbers are not, my figures are not exactly the same as Brash Singh showed this morning. We both agree, and I think everybody, that there is a very active development and growth of the microbiome market in the very, very near future. However, to develop a comprehensive microbiome knowledge base in support of sustainable agriculture and circular economy, we need large-scale, international, multi-year and multidisciplinary effort. And to overcome the barriers of scale, reach sufficient critical mass, share data, and link different disciplines, we propose to elaborate the strategic research and innovation agenda to align priorities, to defragment the microbiome research area, and to promote multidisciplinary and international collaboration and training. This agenda and this field of science and innovation resonate with the consumer demand for healthy, nutritious, and sustainably produced food, need to be centered on the understanding of how microbiome can stimulate or correlate with performance of plant, animal, and human, but also how this knowledge can be translated into innovative application, and should support the validation of proof of concept and emerging business opportunities. So we elaborated, or we benefited from different actions developed in the frame of microbiome support. So for example, we had 3 transsectorial workshops which allow us to identify 5 transversal issues or priorities, which are stakeholder and society awareness and capabilities, open science, data access and sharing, research infrastructure for data but not only, knowledge transfer and innovation, and regulatory science in support to regulation frameworks. For those 5 issues, we kind of make a state of play, define and prioritize gap, and identify mission to fill those gaps and reach the vision. We also use other action to define 5 thematic areas. So we started with a mapping exercise, which identified 257 strategy and policy documents for food and agriculture from 25 countries. And this mapping is available on the Microbiome Support website. But as you see, there is only 30% of those documents which are mentioning microbiome to some extent. Among those documents, we identified 20 national and governmental strategic research and innovation agendas, but only 8, and I have to say only for 5 EU member states, those SRIA mention microbiome. So the analysis of all those documents allowed to identify 5 thematic areas, which are here shown in color box, and below you have sub-priorities. So one of the thematic areas concerns the unlocking of microbiome diversity, knowledge and value, enhancing sustainability and productivity of food and feed production, improving quality and safety of food and feed products, produce healthier food and diets for healthier humans, and expand the microbiome potential for a healthy planet based on improving circularity and adaptation and mitigation of climate change. And you see, yeah, okay. So it's where we are right now. In December, we will have a first draft of strategic research and innovation agenda with those 5 thematic areas and 5 transversal priorities. And in January next year, we are going to start a consultation phase through a Delphi survey, and the opening of the Delphi will be advertised through microbiome support communication channel. This phase is very important to get larger input and feedback and to integrate those comments in the SREA. So to conclude, I would like to say that microbiome science is a young science and it still needs long-term support to advance knowledge. However, it's already contributing to bioeconomy and circularity and to improving food systems, sustainability, safety, and health. There is a fast-growing microbiome market, which is closely connected to this research and innovation field. And in the frame of microbiome support, we started an iterative, multidisciplinary, and collaborative process to harmonize our strategic research and innovation agendas. And in fact, in 2021, we will have 2 consultation phases. The first one that I mentioned already in January. And then after integration of the feedback, a second one, which will be probably more detailed or focused to elaborate with the help of the community, strategic agenda fitting our vision and needs. And to end up, I just want to mention and acknowledge all the people who are contributing to this task, and thank you for your attention.
Thank you, Emmanuel, for your great overview on the development of the research and innovation strategic agenda and the activities in the microbiome support project. I'm sure it's a right kick-off of a lively discussion, and I have the pleasure to hand over to Beatrix Webner from the Austrian Institute of Technology. She will act as the moderator of this discussion.
Thank you, Angela, and thank you, Emmanuel and Yolanda. I'm not sure, Yolanda, are you still there? I know that Yolanda had to leave earlier.
She left already. So.
Okay. Again, like in the morning session, I would like to also encourage you to put forward your questions either in the chat or raise your hand and switch on your camera, and we will invite you in to the discussion. So perhaps something that I would like you all to think about is, and that we could make a round at the end of the discussion. Emmanuel introduced the strategic research and innovation agenda. So what do you think needs to be addressed under all circumstances in this strategic research and innovation agenda? Please also add your thoughts in the chat, or we will address that in a few minutes. I give you some time to think about it. Perhaps to also a question, we have heard a lot about restrictions, and Yolanda also talked about gaps like standards and study models. So there are a lot of different regulations touched when it comes to microbiome. And we heard in the morning that sometimes it takes years to get a registration for a product. How do you think we can overcome this challenge? What barriers do we need to overcome, and how can we tackle this challenge so that it's faster, it's easier? And I see Lena has raised her.
Yeah, I have all. Thank you. I already put something in the chat, but I would like to just here to mention that it was nicely put in Emmanuel's description that we need R&D in regulatory. I very much support this. But it's also important that we don't only see R&D to support the current regulatory. We need obstacles to be removed. There is an ancient, carried over from history, the last decades, because of safety. You have looked at anything which could be called waste. You can never use for anything you call food or feed. And there are many things where we can see it could be healthy food, which is coming from an upgrade of something we discarded before, but which is actually part of a food chain. It's just not the one we normally eat. So that's one thing. The other thing is enzyme discovery. I tried it myself to come through with regulatory when I was in Novozymes. New enzymes discovered from microbiome. You risk that much of them, it's difficult to identify the origin exactly to the species with some full certainty because there are so many unknowns, for instance, in the termite gut. And that meant that if I couldn't describe where it came from, I couldn't use it. Can you see? So I think we need to discuss, not only take all regulatory for granted and then that they are right, they should just speed up, but we should also challenge.
Yeah. Yes.
Angela, I think, and Emmanuel, you want to reply to this?
Yeah, we had, I just want to briefly report that we had the last 2 days, we had very good workshops organized by Emmanuel and Yolanda on transsectorial workshop on more on the regulatory side. And I think there was a very interesting aspect came out, or at least it was for me. It's maybe not so new, but I think it became for me very clear that this is needed. I think there is too little communication and awareness of, so they operate regulatory bodies and especially the academic sector, they work or they operate in very different areas. They are not connected, they don't talk to each other, so they're not interested in each other, maybe. I don't know, but I think here we need definitely more interaction. So in terms of students and researchers being more aware and in contact, of maybe the thinking, how they have to think regulatory bodies. I mean, there's a certain rationale behind these kind of decisions and rules, and on the other hand, maybe we have to involve also regulatory bodies more in the challenges and the problems and the potential and so forth of of research and innovation pipelines or developments and so forth. So I think we have to learn from each other, and maybe this would improve or help solving one or the other issue.
Yeah, I think it's thank you, Lena, for your great question and your comment. I think it's very interesting. We've seen, it was striking for me in this workshop. We heard scientists talking about release of specific microorganisms like probiotics, like in the environment. And it was quite clear for them that they will never survive, and that there were not so much issue about that. And when they were discussing about that, I was really thinking on the regulation aspect and thinking no way that this will be so easily accepted as an answer. So I fully agree with you that we have to challenge. But it's interesting that you mentioned this experience with enzyme of unknown origin, because I think it also shows that when you want to push the regulatory agency, it's not always you. I mean, it's not always scientists, or even if you have good arguments, which is a winner at the end. So I fully agree with Angela's comment. I think it's really time to work together. maybe not all the time, but to identify actions, to identify priorities on which we need to talk, we need to know each other, and we have to build more confidence and trust between those two type of disciplines, I would say. And otherwise, I think we will continue providing more information and providing more data, and I'm not sure how long it will take to be successful. Yeah. So yeah, I think it's really a challenge to have this multidisciplinary and trainings and communication. Yeah.
Thank you, Emmanuel and Angela. I have another question from Adrian. He asks if somebody has undertaken a comparative assessment of regulatory frameworks at the international level.
Not to my knowledge. We have, but I mean, I'm working in the field of human microbiome and food microbiome, and I know that for human microbiome, United States are a little bit more advanced than Europe. So I think that there is actually startups and SMEs which are choosing to do their trials in USA instead of Europe. And I think that regulation is part of the question. But I don't know for plants or biopesticide. Yes.
I mean, I don't know whether there are comparisons have been done, but I think it's well known that when it comes, for instance, to biopesticides or more the crop production sector, then the registration of products is much faster in the US. And of course, there must be many more regulations worldwide existing, and I don't have the information on those, for instance.
But I think it's a very good point. It would be very interesting to build on that, I think.
Yeah, absolutely.
Lini, yeah.
Can I just add, there are a lot of international, different approaches internationally, and also some knowledge about practices and so on. FDA is opening doors for so many regulatory approvals in other parts of the world and so on. But also the other way. I was a program head at International Rice Research Institute, and there I was studying the submerged microbes around the submerged rice. And there I was simply so appalled to see that it was an attempt to recognize, to approve based on data from southern part of the United States, where you have dry seeded rice, or then directly for having such kind of different types of pesticides used in submerged rice in Asia. I mean, I am talking about that we should challenge, but I'm also very much we have to be so sure we don't do mistakes. There are ample examples of beneficial insects introduced for biocontrol in Africa, which have become serious hazards, serious.
Yes, thank you. We have another question about the integration of human and health regulatory issues under the One Health framework. What about this integration under the One Health framework? Do we have an expert on the One Health framework?
One Health sounds so fantastic, but we don't know everything.
Yeah, I think it's really a challenge in the field right now, which is that, for example, for those microbiome, I mean, food system microbiome, it developed an holistic vision and analysis, and you see that. So it's not siloed anymore. You have to consider the whole system, and you have to consider the gene and microbial flux in the system and communication between the different areas of the system. And I think it's really, it's challenging, it challenges a little bit everything, not only science, but also regulations. Because you cannot consider that, you have to consider the impact of a product, not only in one area, but much more globally in the system. Yeah. So one health is very good, but I mean, it's very interesting. And I think it's very relevant to consider that our health are dependent from different factors. But I mean, we human, of course, we are influenced by animal health and by environmental health. And the actual pandemic shows us how much, and about food safety. But on the other hand, scientifically, it's difficult to build those links between the different areas. And I'm quite sure that for regulation, it will be even more difficult. But maybe in the audience, there is someone who would like to comment on that. Do we have any One Health expert?
Hands up. No? Then I would like to address another question that I think is quite interesting. There is a trend of homemade fermented foods, and they are, of course, not at all regulated or certified. Are there any insights for this issue? We are talking about that we want to we need reference materials, we need standards, but how about all these homemade business trends that have already started? Any insights for this issue, also from the audience?
I think it's a very good question. And much of it will not go through regulatory if you tried. Yes. And there are risks which they don't foresee. So I rest my case here, but it's fun.
I think in Europe for fermented food, we have this QPS microorganisms, which is amazing because it has been defined based on cultivation of isolation of microorganisms. And we see now using metagenomics, for example, that most of the traditional fermented product contain much more than what we thought in terms of diversity and species and bacteria and yeast and fungi and okay, because there is this safe use history. You can try to get all those microorganisms on the QPS list, but even if it takes some time. But it's the same. We are doing the characterization with traditional African fermented product, and we have lots of microorganisms which are there. And those people, they are consuming those products, and it's not a problem for most of the time. It's even nutritious food, but if it should be regulated now, I don't know how it will end up.
Angela, you want to add something to this?
I want to add. I mean, I think generally this raises the question whether we should evaluate the safety and efficacy and functions of a complex microbiome, like the edit functions of each individual strain, which is currently done and which follows this concept of this list of safe organisms, and which also causes which hinders the registration, of course, of many products, because it's far too expensive to do this. But it's also, from a scientific point of view, it raises the question whether this is useful. I mean, can we really evaluate a product based on the sum of its individual components? maybe in some cases, yes, but when it comes to clear pathogens. But in many cases, I think it would be much better to evaluate a complex product as a total unit, and not split it into individual components. But of course, that's a it has of course also some disadvantages and risks. Yeah, but
yeah, I think it's one of I fully agree on that, but I think it's still one scientific challenge to understand how these microorganisms are interacting. So it's very and what are the emerging properties emerging from those interactions. But I think it's a very, very interesting scientific question, and it's quite clear that there is properties. For example, I have colleagues who are always telling me that if we try to put a new microorganism in an healthy ecosystem, you can forget about it. It will never invade the ecosystem. And this kind of properties and how does it work is not so known today, And I'm always giving this example about yogurt, where you just have two species, but there is still paper today explaining how the cooperation between the two species, what are the basis of the cooperation between the two species. So I think we have lots of interesting discovery, but also work in front of us for that. Associated to that, there is really the challenge and the question of ecological engineering, ecosystem engineering, because this is really challenging. We still see that there is a difference between the system, for example, for waste treatment of upscaling. There is a difference with the system we can imagine and what we can find in nature. And really, it will require to understand those functional properties and complementarity, and to be able to have a hand on that. Yeah, I see that Lene would like to.
We have 5 more minutes, and so we have one quick remark from Lene, then I have a very short question, and I think we still have to have our rapporteur Tanya giving a short summary of the session. So very short, Lene.
Emmanuel, you approach something really interesting about this convergence, which happens in many consortia, also shown in anaerobic digestion, but also for food, soy sauce, consortia, fermented meat, and so on. It is amazing how stable it will end up in the same consortia all over the world. It's amazing.
Thank you.
Fantastic.
So one idea came up from Jennifer saying that the FAO microbiome learning pathway, during that, the idea came up that the FAO could play a connecting role for scientists and regulatory bodies. What do you think of that? Very, very brief answer.
Great point.
Good. That was very brief.
Fully agree.
And the other very, very quick question that I have, Emmanuel and Angela, you mentioned the Delphi survey. How can people participate in that? How can they, if they are interested in it and they are not on your list of stakeholders yet, how will this be published?
Sorry,
shall people who want to take part in the Delphi survey to when it comes to the strategic research and innovation agenda, shall they contact you to be put on the list of experts, or how will this work? How can people participate?
Yes, of course. Yeah,
we have not rolled it out yet completely, but there's always the possibility to contact us. But I for sure will also put up something on our web page, right, Emmanuel?
Yes.
So that I think there will be also, it will be also possible to to provide input via the web page, or to get access to that. Yeah. So I think there are multiple ways, yeah, to be integrated.
Yeah.
Thank you. So I think at this point I have to ask Tanya to give a short summary of what was discussed today, and then I think we have to leave for the plenary at 5, if my clock works all right.
So thank you, Beatrix, and also thanks to the excellent speakers also in the afternoon. What I liked the best was that is what was already discussed in the morning, the need to take the holistic approach, and holistic consideration was confirmed also in the afternoon session. And I think the focus of this session could be really put on this regulatory. What are the challenges? What are the opportunities? There are a lot of challenges that I noted. There's this difference in the international standards, regulatory standards landscape. There's also difference in what's done. But what we also see as an opportunity, first of all, to establish better exchange between science and the regulatory, and better dialogue. As Lena said it really nicely, we really need to establish a strong dialogue in order to go forward together. We cannot just expect other side to do something for us. And what I found interesting, even though it's a challenge, but I also see it as an opportunity to include this interdisciplinarity that Emmanuel was mentioning. And also, if we recall how Yolanda nicely compared how actually similar the systems are. So we really need to, I think, break out of these silos and say a rhizosphere for the plant is something that's really similar to gut microbiota for the human health. So perhaps regulatory could also converge a little bit and sort of make it more standard and more easy and more uniform for all of us to go forward. So this would be my key messages that I took from this session.
Good, then I think we really have to come to an end. Unfortunately, it was a very lively and good discussion. There are still some more aspects which we couldn't address, so please apologize for that. I would like to thank joining this breakout session. I would like to thank all participants in the discussion, all speakers, and Beatrix and Tanya for the moderation and for the summary. I hope you have time to all move now to using the other tool again to enter the plenary.